Can you hear me? No? Hello, hello. Guys hear me? Kind of, not that great. Yeah, is that good? Yeah, okay. So it's not quite 2:30 yet, but I just have a few announcements to make first. Hopefully most of you have seen or it's been passed around the evaluation sheet. If you don't have one, there's extras floating around. So if you have extras, could you hold them up so I know where they are? Where are the extra evaluation sheets? I gave it in each section. So there's some there. There should be some in each section. So if you didn't get one of the, it's a regular sheet of paper evaluation sheet, please get one and fill that out. And in the top right-hand corner, you'll see the session number you have to fill in. That's 80. That's what you put at the top right corner there. And let's see what else. Okay, so the flow of this is going to be, we'll start with these questions at the end of the review material I gave you. And we'll go through that. And then if there's time left over, I printed out the exam keys from your exams this semester. Highlighted some questions I thought that are really representative of what you'll probably see on Monday, and or questions that I got lots of questions about. And I'm using version A in all of the exam keys. So if you want to write down the questions or if you have those with you, if we have time, that'll be what we do next. I do have one or a couple corrections in some of the material. So in the main material that precedes the questions, in chapter 10, I flip-flopped what the purines and pyrimidines were in the review. So always remember cut pi. CUT are the pyrimidines, A and G purines. That's flipped in the review. And all that material is just for your benefit. There's not really a whole lot to go through unless you want me to read it to you, which you probably don't. And then there are a few questions that I think I needed to clarify, but we'll get to that when we do it. And it is now 2:30, so we can begin the practice questions. Everybody good? Any questions before we begin? Yeah? Where's what? It's on the SARC site. You go to the UCF SARC SI, you go to the Study Union Review Materials, and you'll see your class number and my name, which is Emory, PCB 3063 Emory. You can ask people around you if they found it. They know where it is. Okay, so this is in chronological order, so starting from chapter 2. I did focus more on the earlier chapters because you probably know you had a really big test a week or so ago, and that was over 6 chapters. So hopefully that's still fresh in your mind, and we can go back and work with some of the newer or the older stuff. Okay, so let me get caught up again. Okay, so this is not a difference. So these kind of questions sometimes can be difficult. Not the difference between prokaryotes and eukaryotes. The answer to this question would be D. Eukaryotes do not undergo binary fission. Everything else is true. Okay, and if you ever can't see the question on the screen, let me know. And the 3 parts of eukaryotic, here we go, that's the key word there, chromosome structure. The answer to this would be C. You need telomeres, centromeres, and an origin of replication. Those are the 3 main parts. Okay, and here's the first question that kind of need a clarification. This word shouldn't have been telomere. It should be centromere. But you probably figured that out. Okay, so what best describes the centromere location that is almost to the end of the chromosome, so almost to the telomeres? The answer to that would be D, acrocentric. E isn't anything, that's just a made-up word. So make sure you know all of these 4. A through D are real locations. Okay, let's see. Okay, and here's probably the stuff that you guys hate the most from chapter 2 is counting chromosomes. So I'll draw it out here. A cell in G1 of mitosis has 8 chromosomes. So that means that's the 2n number. How many chromosomes and DNA molecules are in anaphase? So if you go ahead and draw it out, what you'll see, probably it's easier to visualize metaphase before you go into anaphase. So, and remember when you're counting chromosomes, you're actually counting the centromeres. So that's 1, 2, 3, 4, 5, 6, 7, fix one in there, 8. So you'll see 8 centromeres, which means there's 8 chromosomes here, and this is metaphase. Then next we're going to go to anaphase. And these are now going to get pulled apart. Anaphase, you can think starts with A. Anaphase, are moving them away from each other. So then you're going to get 1, 2, 3, 4, 5, 8, 9, 10, 15, squeeze it in there, 16. So now if you count the chromosomes again, each one now has its own centromere. So you double the amount of chromosomes and the amount of DNA. So you go from 8 to 16 chromosomes and 16 DNA molecules. That's almost, I truly believe, it's safe to say that in anaphase, usually you're going to be doubling. That does change a little bit in meiosis. Okay, and you can see, yes. Okay, so the same kind of thing again. Your 2n number is 12, so how many chromosomes and molecules are there at the end of meiosis 1? Meiosis 1, remember, you're going from your diploid number to your haploid number. So here, we would have 6 chromosomes because we're now haploid. But remember, those chromosomes are still in the homologous pair type of form. They're not in their chromatid form yet. That's what happens in meiosis 2. Okay, all these wires are in my way. Okay, and now we're looking at what gametes, so make sure you know that vocab word, what gametes a parent with the genotype homozygous or heterozygous would make. And remember, when you're using gametes, you want the haploid, so you want one of each gene or each characteristic. So let's see, you can immediately eliminate things like D because you have both E's in one gamete, which would be something that would say that something went wrong. So D and C both have gametes that have both traits in them. We don't want that. And then you have a heterozygote. Heterozygotes offer the most variety in gametes, so your answer here would be E. Okay? Let's see if I can make this better. Okay, so you have these parents. I'm not going to name them. And then what is the probability of having this type of child? Let's see. Okay, so this is a probability question. I would not recommend doing a Punnett square for this. So break it up into each letter. So your AA, the odds of your first parent here giving a big A is 1/2. The odds of the second parent here giving a big A is 1. So you multiply that because of your and or rule. So this would be the A gene. Then you look at your B gene. The odds of your first parent giving you a big B is again 1. The odds of your second parent giving you a big B is 1/2. So now this is your B probability. Now we are looking at the C. So the odds of getting a big C from parent 1 is 1. The odds of getting a big C from parent 2 is 1/2. So here is your C. And now D. Let's see. Okay, so the odds of getting a little d from, let's see, parent number 1 would be 1. And the odds of getting the big D from parent 2 would be 1. So we know that each of these has to come from them. And let's see, now we're moving on to E. So big E's, that's 1/2 from the first parent. And then only one thing can be given from the second parent, so that would be 1 again. And then the last one here is F, and we're doing heterozygote F. Be a 1/2 chance here from the first parent, and a 1, because it can only give 1 big F here. You multiply all of these 1/2s together, and you should get 1/32. Okay. Okay, and here is your basic Punnett square rule, or you could make up letters and actually do a Punnett. If you know your rules, then you should this should be a fast problem for you, but if not, this Punnett isn't that hard. So any letters, I always recommend using a letter that's really easy to tell capital from lowercase. Don't use O's, S's, that can get confused very easily. So you're crossing heterozygous parents, so let's use A's, big A, little a, big A, and little a, and just fill your Punnett. And then you're looking for the probability of having a homozygous dominant, a heterozygote, and a homozygous recessive. So again, you might remember these rules, but if you don't, you'll have a 1/2 chance of a heterozygote, 1/4 chance of a homozygous dominant, and a 1/4 chance of a homozygous recessive. And these correspond to the order that it was given in the question, so the answer should be E. Okay, and here is a straight memorization type question. The mode of sex determination in which females are the heterogametic sex. Remember, hetero meaning different, and this would be D. And always use these questions if you don't remember what each of these are determining for. Go back and check that out. Okay, fill in the blank, and inactivated X is called a what and is responsible for what. So we should remember inactivated X is a bar body, so you can eliminate the ones that don't offer that as an option, and now is responsible for dosage compensation. Turner syndrome is something completely different, although it does involve the X chromosome. And a 2-to-1 ratio in a typical Mendelian cross indicates there is a lethal allele present. If you don't remember all of these ratios, I would definitely go back and check them out, put them on flashcards, something, because it makes questions like these much easier if you remember that. Okay, now let's see. So we have this cross. There are 3 phenotypes presented. That should kind of raise a red flag if there's 3 phenotypes and we only have 2 genes here. The genotype EE, so homozygous recessive, is present, and when that's present, the color is always the same, regardless of whether this B is big or little. And what would explain this phenomenon? This should recognize this is an epistasis question, and we are talking about a homozygous recessive gene acting. So you should think, okay, so it is recessive epistasis. It's not going to be dominant, so you can eliminate B and E. So now you just need to figure out if A is true. A is saying that the B gene is the hypostatic gene, and the E gene is the epistatic gene, which is true. The epistatic gene is the gene that is influencing, silencing some sort of influence of the other gene. So both A and C are true. Okay, and here into pedigrees. In this mode of inheritance, males cannot pass on to their sons, so that could already eliminate one option for you. And females pass on to half and half. So we know it's not Y-linked if males can't pass it on. And when there is a difference between the sex ratios, you know it's going to be X-linked. So you can get rid of the autosomal as well. Now you're left with dominant or recessive, and the answer here would be dominant because of the female passing it on to half and half. And definitely go back and memorize those rules for each pedigree if you haven't already. Another pedigree type question. If the daughter does not have the disease, but the father does, what mode of inheritance can you definitely rule out? So I would recommend drawing something like this out because it's pretty simple. So the daughter does not have it, but the father does. So here's your super simple pedigree. And from there you can figure out some things. You can't rule out Y-linked because you don't know if he has a son and his son has the trait, so you can't rule that one out. Let's see, what else can't you rule out? Or you could do through each of these, try to figure out which one would work, which one wouldn't. The answer here would be you can rule out X-linked dominant because that would be passed on every time if the father was afflicted because he would give his dominant X to his daughter. In the recessive, he might not, or the mother might cancel that out because she's not affected. And so, yes, okay. Okay, and here, there's another clarification. It's a recombination frequency, and those are what the percentages are, just to be clear. So there's 3 genes. Y and Z have a recombination frequency of 20%, X and Z 25, X and Y 5. And you're trying to determine the order. First thing, always do, find the biggest percentage. In this case, it is 25%, and that corresponds to X and Z. So X and Z are going to be the ones that are the farthest apart, and from there you can already eliminate answers. So it's definitely not this one, it is not this one, and it's not that one. So you don't even have to figure out anything else. You just know that that's the answer. Okay, and here you are trying to determine the recombination frequency, and that is a formula that definitely I would expect to see on the final. So you just take your recombinants, divide them over your total progeny, multiply by 100. So in this case, you have 110 total. Don't assume you're always just going to have 100. You have 24 recombination progeny, and you have 86 parental. Not super important, but just for information. So you just take 24 divided by 110, multiply by 100, and you should get 21.8%. Okay, and here's another way of determining gene order. You can do so via the double crossover. So if you know a double crossover took place and produced these offspring, what is the order? From here, you always use your preschool rule of what doesn't belong. So here, we have little, big, big. So little is the one that stands out. We have big, little, little. Big is the one that stands out. The gene that is not in line with the other ones is the one that's in the middle. So you're looking for E in the middle corresponding to answer A. Another way this question could be asked would be the offspring that is produced in the least amount. That also corresponds because double crossovers are the rarest offspring. Okay, and a loss of a homologous pair, whenever you see homologous pair, always think you're talking about 2. So your 2n minus or plus is always going to involve 2. So you can immediately get rid of the ones that involve 1. And then from there, you need to remember what your different words here mean. So monosomy, nullisomy, trisomy, each of those correspond to a, yes? Number 17? Okay. Okay, so here, let me kind of, okay, so here were your offspring, and you use your rule that you learned in preschool about what doesn't belong in this order. In this case, you have a little e, a big F, and a big G. So this little e is the one that doesn't belong here. In this one, you have a big E, little f, little g. So this big E is the one that doesn't belong, which means that's the one that got crossed over twice, which means that's one that had to be in the middle of your gene. So you just find the one that doesn't belong. That's the one you know is in the middle of your gene order. Yeah. Okay, so back to 18. Each of these are just vocab you have to know, making sure that each one of these does correspond to the proper number here, because obviously not all of these are involved in 2n minus 1. The correct answer here is B. Okay, so you have a haploid number of 9, and how many chromosomes are in a cell that underwent a monosomy? So this question is definitely, I think, tricky because you're given the haploid number, and whenever you're dealing with any of these non-disjunction, you want to work with the diploid number. So make sure you change this into the diploid number, which would be 18. And a monosomy is a minus 2n minus 1 relation, so you're looking for 17. Get crooked. Okay. And here now we're in population genetics with Hardy-Weinberg. So we have 40% homozygous dominant, 20% heterozygous, 40% homozygous recessive. You're looking for the frequency of P and Q. Remembering that P usually corresponds to the dominant, P the recessive. And remember here, P and Q are in their singular form, and you're given them in their genotypic forms. So you're going to have to eliminate some of them. So using some of your equations that you should remember, we can determine that P is equal to B, and B is 40% homozygous dominant, so all of these big B's are going to correspond to P. And then big B is also present in the heterozygote, but it's only there in half of the genotype, you could say. So 1/2 of 0.2. And Q turns out to be the exact same thing. 0.4 would be homozygous recessive, plus half of the heterozygote. And when you do this, you should end up getting this, A, half and half. Okay, another type of equation. We have population of butterflies, 15% are blue. Blue is recessive to yellow. So what is the frequency of heterozygotes in the population? So we're given Q in this situation. And Q squared equals 0.15, the decimal equivalent here. And we want Q, so you take the square root of that, which is going to be equal to 0.387. We know that we need the frequency of P as well, because we're looking for the heterozygote. So always remember P plus Q has to equal 1. So if we say that P has to equal 1 minus 0.387, that is going to give us, can you see? Yeah, 0.613. And you're not done yet. You need to find the frequency of the heterozygote. So then what you will do is you'll have to remember here that that would be 2pq. That's the Hardy-Weinberg equivalent of your heterozygote. So you'll multiply 2 times 0.387 times 0.613. You do that math, you should get B, 47.5%. You just need to look at it again? Yeah? Mm-hmm. Okay, So which of the following decreases genetic variation within populations? I included this chart into the information that's before all these questions. I would know every single factor that influences, what happens in populations, within and in between. So remember, keywords here, you're decreasing within. So the only one here that decreases within would be genetic drift. Remember, genetic drift, gene flow, migration, not the same thing. Migration and gene flow are the same thing. Genetic drift, something different. Okay, so in 23, we're working with DNA, and the strand is 18% adenine, and from that, you can determine the percentages of the other bases. A, we know, always bonds with 2 bonds to T, so 18% of adenine should correspond to 18% thymine as well. So you can get rid of the ones that don't have T having 18%. And now you have 50-50 chance if you don't know what else to do. But from there, you can remember that G always bonds to C, so G and C should have the same amount, and everything should equal 100%. So you find that out. This equals 100%, the other one does not. That has to be your answer. Okay, and here is your scenario. A new organism, and you extract its DNA. In the lab, you notice that the DNA is left-handed. So what type of DNA does this organism possess? The answer here would be Z DNA. Remember, we have B DNA. That involves the DNA in water. A DNA usually does not like water. And H DNA, I think, is 3 strands wrapped around each other. Okay. So facultative heterochromatin. Remember, facultative, you can think, can go either way. Constitutive, always going to be the same thing. Not going to change. So the only answer here would be the X inactivation, because it could be one, could be the other, could change, might not. Telomeres, centromeres are always going to be condensed. Okay, so here is vocab corresponding to some concepts. How many histones are in the nucleosome, and how many total would there be in the chromatosome? So remember, the nucleosome is what the DNA is actually wrapped around, and that's an octet. And so you have 8 in here, but the question is asking for different histones. You have an octet, and each one of them corresponds to 2 of its copy. So you have 4 total here, because each of them has 2. So get rid of any answer that doesn't have 4 in the beginning. And then total in the chromatosome is going to be your 8 number plus your 1 histone that isn't involved in the nucleosome. That is going to be then equal to 9. So your answer should be C. Okay. So if DNA polymerase was not functioning during replication, what would happen? And this is the key here, DNA polymerase 1. Know the difference between 1 and 3. Okay, so DNA polymerase 1, does that involve nucleotides or does that add nucleotides to the primer? No, that's DNA polymerase 3 that does that. Would there be residual RNA after replication? Yes, because remember, DNA polymerase 1 is the one that comes in, replaces those RNA nucleotides with DNA nucleotides. And it also has error fixing capabilities, so yes, this is also true. So only B and C. Okay, and how many primers are needed on a leading strand and lagging strand with 42 Okazaki fragments? Here, remember, the leading strand is continuous. So once that 1 primer is in place, it can keep going and keep going. So you only need 1 primer for the leading strand, but you need a primer for each lagging strand fragment. So okay, so this is kind of similar to a clicker question that you saw. I do tell you that this is RNA, but you should be able to figure that out because U's are involved. And the question is asking whether it is single or double-stranded. So remembering your base pair rules, A to U, and G to C. These do correspond. They do have the same number, so that means that this will be double-stranded. Okay, and here you're given the RNA molecule, and you are asked to provide the non-template DNA strand. So from here you can remember the simple rule that you just need to change your U's to T's. And this question is a little bit different because I didn't give you it in the correct, I guess, flipping way. So the correct answer here is D, I believe. Let me check to make sure I tell you the right thing. Yes, D. And it might be kind of confusing because I flipped it, but all that needs to have is the correct directionality. So 5' to 5', you have C, G, and then change this U to a T, A, G, A, and change this U to a T. This would be the correct sequence. Remember, DNA exists in 3 dimensions, so it can flip and move. You can read it forwards, backwards, but as long as it has the proper directionality, that's the correct answer. Okay, and here we're talking about the operon, so the lac operon specifically. Let's see, the regulator protein in the lac operon is either going to be an activator or a repressor, and that is going to be something, and yeah, that's kind of how it's going to go. So the regulator protein in the lac operon is a repressor, and let's see, allolactose is the inducer. So remember, the lac operon is a negative inducible operon. That negative always corresponds to a repressor. When a positive something operon is going to correspond to an activator. So remembering lac operon is negative inducible, you're going to have negative being the repressor. Remembering that allolactose acts as the inducer to turn on this operon. Okay, so glucose levels are high. What will that do to CAMP levels and lac operon transcription? So remembering that these things are inversely related. So if glucose levels are high, you don't need the lac operon to be on because your body, and especially E. coli, wants to use glucose over anything else. So that means that it will decrease. So we're not going to have increased anything. So we will have decreased CAP and CAMP. CAP and CAMP is what helps to turn on the lac operon. So if we have more glucose, which means less CAMP, there will also be less transcription. It should be B. Okay, and here's asking what increases transcription rates. And remembering that the hats here are involved in acetyl groups. Sulforaphane compounds are silencing HDAC, so transcription will take place. That way, this question will be E. HDACs take away the acetyl groups. Definitely know your relation between methyl and acetyl groups. Okay, so you're given the correct amino acid sequence here, and you have a mutation that results in this, and it's asking you what most likely happened here. So going through here, a nonsense mutation, that means that something would stop, and actually the opposite happened. We have a longer protein, so that's not what happened. A codon insertion, you can see everything is correct up to this point. Then all of these amino acids are different or new. So a codon insertion wouldn't cause a frame shift and cause everything to change. It would just either probably insert somewhere. So there wouldn't be any frame shift if it was a codon insertion. Okay, and I'll skip down here. Silent mutation, you wouldn't be able to tell. So definitely that's not what happened. So you would end up getting the same amino acid if you had a silent mutation. A substitution, again, wouldn't result in a whole frame shift and change all of the preceding amino acids. So your answer here is base deletion. You delete that one amino acid that codes for proline. It shifts everything over. So now your entire frame is reading the wrong thing. Okay, and now we're talking here the first tRNA in translation. You have your 3 sites. Probably you know it's not going to be the E site, but your first tRNA is going to bind to the P site in translation. That's the exception to the rule there. All other tRNAs are going to bind to this A site. Okay, and back to the operon. Okay, so permease, one of your genes the lac operon codes for, is coded for by which gene? So another name for the permease gene, that will be C, the lac-Y gene. Definitely know what all of these genes are coded for. I don't think lac-P is a real gene. I think I made that up. Okay, so you're given a positive inducible operon. So again, you're talking about positive, and we just talked about how positive involves a regulator gene that is an activator. So you can immediately eliminate your repressors. And the inducer, that's not a regulator gene. And in a positive inducible, is your activator in an inactive or active form? So remember, inducible then corresponds to being usually off. So So if this operon is usually off, but you have an activator that the regulatory gene codes for, that means that activator is usually going to be inactive. Something else has to come in, make it active to turn on transcription. Okay, so not an example of a transversion. Remember, a transversion would be a purine to a pyrimidine or a pyrimidine to a purine. So not an example. So this one is an example, and this is an example, and here is your not example. Okay. Okay, so you know our mutation took place, and the mutation changed an asparagine for an asparagine. That is an example of a silent mutation. The codon changed, but it didn't change the amino acid. This usually happens in the third position, so also known as the wobble position of the codon. Okay, and so now you're asked about a repair mechanism. In which repair mechanism are nucleotides replaced and not repaired? So in mismatch, we're talking about repairing, so this is incorrect. And you can think base excision. Your excision means to take out, so that's probably going to be it. Direct repair also does take out. Okay, and then here you're talking about the biotechnology. So what molecules are studied in southern, western, northern? Southern, always DNA, so get rid of your ones that don't start with DNA. Your western, going to be protein. Northern will be RNA. Here. Okay, so these are the questions that I made up. Remember, your exam will be 100 multiple choice questions. So you could have tried timing yourself with this, see how you're feeling with that kind of regard to anything. Do you guys want to see any questions again before I move on to the old exams? Good, any questions about anything? Any questions? Yeah, go ahead. 26? Okay, so here, did you want me to explain it, or you just want to see it again? Yeah, sure. Okay, so different histones in the nucleosome. You know in the nucleosome there are 8 total, and that would be your H2A, H2B, H3, and H4. That's what's in your nucleosome. But you're going to have 2 of each of these to make your octet. So different histones, you're only going to have 4. You'll have 8 total, but only 4 different. In your chromosome, you're going to have, that's the entire complex. So that would be, let's see, your H1. So total would be the 8 here, plus your 1 H1. That would give you a total of 9. Anything else you want to explain in more detail or question you want? Yeah. 38. Sure. Okay, so this is a definitely straight up vocab type question. So a transversion is going to be a purine to a pyrimidine or vice versa. Okay, and the other one is a purine to a purine, pyrimidine to a pyrimidine. So you're looking for what is not an example of this. So what would be a transition? So you're looking for the transition example. An A to a T is a purine to a pyrimidine, so that is a transversion. A G to a C is a purine to a pyrimidine, that is a transversion. T to a G is a pyrimidine to a purine, transversion. A to C, purine to a pyrimidine. So the only example of this is C to T. Remember cut pi? Both of these are pyrimidines. So they will be not a transversion, but a transition. Yeah? Okay, so these were your blotting techniques. Your first one discovered was your Southern, and that was by an actual Dr. Southern. So that's kind of how I remembered it, is that the Southern technique was the first one discovered. DNA is kind of the most important molecule, arguably. So that one, Southern, the first one discovered, has to be DNA. Western and Northern then, I'm not sure you can come up with your own type of ways of remembering it, but each of these blots corresponds to these types of molecules. Southern to DNA, Western to protein, Northern to RNA. Anything else? Any questions about the material before all of these questions? No? Okay, so next I will move to the old exam, starting with the first old exam. And I will let you know which questions that I have the printout, but I'll let you know which questions that I'm using. If you didn't bring it down or bring it with you, you can write it down. And, okay. If anything comes up, just let me know and I can explain it. I might need to zoom out. Okay, so all of my versions I'm using here are the A version. And all of these questions either I think are representative of the broad things you'll have to know, as well as questions I got lots of questions about. Okay, so this is the first question in exam 1A. Okay, so a cross of 4 independently assorting genes. Another good technique is being able to root through all of the irrelevant information and find the information that really matters. So all that really matters here is you have 4 independently assorting. You have your genotypes of parent 1 and 2. And then the question is asking the chance that just one of their offspring will have the same genotype as parent 2 given here. So it doesn't really matter, same genotype as parent 2, just the odds of getting this genotype. Okay, so here your genotype would be little a, little a. Parent number 1 only has big A's. It can't give a little a. So right there, you know that that's impossible. So you can go with 0 right off the bat, or you can keep going, and there's even more things wrong with this. Parent 1 also only has big B's, so there's no way that their child can have homozygous recessive for B. So again, impossible. So your answer would be 0% chance, 0 children will have this. Yep. To use for what? Just in general, just this type of problem? Okay, so let me get a piece of paper just so you can see it a little better. Okay, so you're trying to find you're crossing these two in hopes of getting this offspring. And remember, these are gonna, each of these parents are gonna produce gametes, so those gametes are gonna fuse to produce this offspring. So only half of each gene will be in the gamete. So parent 1 would produce 1 gene, or 1 gamete that would have A's, but any gamete it produces is only gonna have big A's. So let's say these are its gametes, and it's gonna fuse with the gametes of the other parent. No matter what happens, here, you're going to have a heterozygote. So there's no way that this child could be little a, given this genotype of the parent. Same thing with B. Can't happen. This one only has big B's. Can't donate a little b. Okay, so I'm skipping through some of these. Okay, so the third question here. You're given 2 people, you know they're both heterozygous for an autosomal recessive disease. Their firstborn child has sickle cell, and the mother is pregnant again with another child. So what is the chance that both of those children will have sickle cell? So here, you just do a Punnett square. Let's see. So you know they're heterozygous. So again, use something that's easy to determine uppercase versus lowercase. And it is an autosomal recessive disorder. So if you know your ratios and your fractions, you know to get little a is going to be a 1/4 chance. But you want to know the chance both. So remember your and or rules. Both of these children having sickle cell, you're going to multiply. 1/4 by 1/4 is going to give a 1/16 chance that she will have 2 children that have sickle cell. So again, you could have done a Punnett here, or just start to remember what these basic Punnets produce. Yes, do you know the and or rule? Can we explain that? Yeah? Okay, sure. Let's see. Okay, so and or sometimes also either. So the and rule, and this is used in statistics a lot, is when you're given something with and, you're going to multiply. When you're giving a problem that has or either, you're going to add. So here in this question, you're looking for child 1 and child 2 having sickle cell, so you'll multiply. If it said child 1 or child 2, that would change the question. But sometimes this is, because again, it doesn't explicitly say and, sometimes you have to assume that it's and. But in this question, is and multiply to get 1/16. Yeah. That is the question that I got a lot, and this question, I think, is asking the chance that both children will have sickle cell, kind of assuming that you don't know that the child already has sickle cell, because the correct answer is 1/16, according to her key here. And it's just looking, I guess, the wording might be a little ambiguous, that child 1 and 2. So again, you're just going to multiply the probability that either of them is going to get it. So this is a question that got a lot of questions on. I think it's because lots of people picked 1/4 instead of 1/16. But it's the probability of both children, both meaning the multiplication rule. Yeah? Good? Okay. So now number 6 on exam 1 version A. Okay, so recombinant chromosomes are created during crossing over events between what? So remember here, crossing over recombination only is going to happen in meiosis. So some of these options give you mitosis. That's definitely not going to be right. So now what you're left with figuring out is if it's going to occur between non-sister or sister chromatids. It has to occur between non-sister chromatids because sister chromatids are identical. So if sister chromatids swap information, you wouldn't even know that they swapped any information because they're the exact same thing. So it has to be B, non-sister chromatids in meiosis. Make sense? Yeah? Okay. Okay, so now number 8 on exam 1 version A. We have an X-linked recessive disease, and everything else kind of doesn't matter. What would be the genotypes of the parents of a female with this disease? Okay, so we know it's X-linked recessive, so if this female does have the disease, she has to be little d, which means her father had to give her a little d because he can only give her one X. That one X had to have a little d attached to it, which made this problem pretty simple in that there's only one father that has a little d on it, on the X chromosome, so it has to be A. All the other fathers have big D. You could also find out the mother, but in this kind of situation you don't have to because all your other options are eliminated. So make sure you go back and check your answers as you're doing these problems. Sometimes they can be done faster just by eliminating things versus trying to just find the one right answer. Okay. So this is number 11 on version 1A of exam 1. Okay, so a human with XXY sex chromosomes would have a diploid number of what and would be phenotypically what? In this case, it doesn't say anything that the Y chromosome is altered in any way, so you assume that they're male. So you can eliminate A. The diploid number of humans is 46. This person is going to have that extra X chromosome, so 46 plus 1 will be 47 with that Y chromosome making them male. Okay, so this is number 12 in the same exam, same version. Okay, and this is, again, mostly, it's a lot of information, but it's mostly just vocab. So you're given a lot of information, and then, let's see, you are mating a black gerbil of unknown genotype with a brown gerbil. If you go back and look, this brown gerbil was recessive. So it's going to be little b will be brown. So if you're mating an unknown with a recessive, that means you're doing a test cross. So any of these other ones going to be wrong. So you're already at 50-50. And then you do your cross and you get half are brown, half are black. That means that this black gerbil can't be homozygous dominant because if it was, all of the offspring would be black. That can't be right. Has to be able to give that little b to produce brown gerbils. So the answer would be D. You're doing a test cross, unknown versus a homozygous recessive, and the genotype here has to be able to produce brown gerbils. So root through all that information to find out what they're actually asking. Okay, so given you a diploid number of 38, the end of anaphase of mitosis, both of those things important, how many chromosomes are present? So anaphase of mitosis, kind of similar to the question that we did during my practice questions, you're going to double, and then you're going to pull apart. So you're given all of these X's, I'm obviously not going to draw 38 of them, and then they're going to get pulled apart. Once they get pulled apart, you're doubling because now each of these chromosomes has its own centromere. 38 plus 38, or times 2, 76. Okay, so this is now 17. Okay, so this is an X-linked gene coding cat hair color. But it'll either be black or orange. And this is also talking about there being X inactivation, so there can be the tortoiseshell color. So you're mating a homozygous black female. So X will say BL for black. And made it with an orange male. Male only has one X, and we'll give him O for orange. So the expected colors of their kittens. So fill out your Punnett. And let's see, so the mother here would donate a BL, father an O, BL and O again. The mother here can only donate BL, BL. So you're only gonna have here, you have black kittens, and here you're gonna have the tortoise shell. And then you can check your answers, make sure that's valid. C here is 2 black males, 2 tortoise shell females. Okay, this is number 18. She's mentioned blood type a few times in class, so I would expect to see a blood type question on the final. So a mother has type A, but you don't know what type of type A. Could be big A, or AO. And the father has type B. You don't know what kind of type B. Could be BB or BO. So you're just looking for the possible blood types of their children. So you could have definitely AB, A and B could combine. You could get A and B. Or if this is the genotype of both parents, you could end up getting O offspring. So A, you could have any type of blood type of child. You could also do all of these Punnets, although it would take some time. Okay. Let's see. Okay, so this is number 22 of the first exam, version A. And you have a recessive epistatic gene. That's your most important information giving in this problem. So your recessive epistatic gene creates yellow labs, and you have 2 labs mated. These are your 2 labs that are mating. What are the possible coat colors of their puppies? You'll note that both of these labs have little e, so it doesn't matter what those big B's or little b's are doing, all of their puppies are going to be yellow, because this yellow gene is the recessive epistatic gene, creates that yellow color, regardless of what this black allele is doing. Good. Okay, so this is number 24. Pea plants carry 2 copies of the gene for flower color, but their gametes only have 1 copy. This is an example of what? Definitely expect to see law of segregation, law of independent assortment on there, probably the 2 most important concepts in genetics. So C and D are kind of nonsense here. So you're always going to end up being between segregation, independent assortment. Let's see, okay, so the question here, they have 2 copies, but their gametes only have 1. You can think this kind of concept, this vocab definition corresponds to the term segregation, because each of these 2 copies is segregating into each gamete. That would give you the correct answer. Or you can remember that law of independent assortment is talking about how each of these 2, or every allele in a person is going to segregate independent of what the other alleles are doing. So this allele here, that does not care what this allele is doing up here. That's what independent assortment is. This is going to sort no matter what other alleles are doing. There's no coordination. There's no, oh, this gene went over here, I'm going to go to the other side. They just move based on how they line up in metaphase. So that would be independent assortment. So if the question said something like, pea plants carry genes and their genes separate independent of one another, or regardless of what one gene separates, another one separates, that would be independent assortment. But going from 2 to 1, that would be segregation. That makes sense? Kind of. Sometimes it's difficult to get those vocab terms down. Okay, so again here, now we're talking independent assortment, 2 genes. An individual who is homozygous dominant for gene A, but a heterozygote for gene B would produce which of the gametes? So I would definitely draw it out. So homozygous dominant, big A, and a heterozygote for gene B. Oh, I'm sorry, I did this wrong. Oh, no, I did, okay. So heterozygote for gene B, let's see. Yeah, there we go, okay. So here's your A, here's your B. What combination could you get? You can get only big A's here, so any answer that has little a's isn't correct, but you're not given any. This answer wouldn't be correct because remember you want to go to your haploid number. And you can get a big A joining with a big B, or a big A joining with a little b. So the answer here would be B. Okay, and now this is exam 2, version A. Question number 1. So this has to do with your pedigrees and population genetics and all of that stuff. Okay, so I kind of got it started here. So your recessive condition occurs 1 out of every 400 individuals. You're looking for the frequency of the recessive allele. So 1 out of 400 is going to give you your genotype, your little a, which corresponds to, you could think, little a squared. So to find the frequency of just one of those little a's, you're going to take the square root of 1 over 400. That should give you 0.05. Okay, so a form of dwarfism is dominant, autosomal dominant disorder. We have somebody who does have the disorder and a parent who has normal height. So we're trying to find if this person is going to be homozygous dominant or a heterozygote. If she has a mother or a father who has normal height, that means that they had to give Jane this little a, which means she has to be the heterozygote. There's no way she could be homozygous recessive because she has the disorder and the disorder is dominant, and you can determine it based on just the information given. Okay, so you're doing a test cross here. Big A, little a, big B, little b, so a heterozygote, and you're crossing them with what you would expect, a homozygous recessive individual. And you have 4 classes of offspring and equal frequencies, 25, 25, 25, 25, or you can think of it in a ratio term, 1 to 1 to 1 to 1. So what has to be true? Again, I recommend going back and looking at what those ratios were each referencing. And in this case, 1 to 1 to 1 to 1 is either independently assorting. You can get independent assortment by either being on different chromosomes or being so far apart on a chromosome that they're going to recombine every single time. So the answer here is E. They're either on the same chromosome but very far apart, or different chromosomes that are going to shuffle each time. Okay. And evolution here, the keyword evolution, the only thing that really corresponds to evolution we've talked about is natural selection. This has to do with Darwin. There were only a few slides on it, but this is definitely an important concept. So all of these other things have to do with Hardy-Weinberg. Evolution, not super involved. Hardy-Weinberg, that's Darwin's thing. Natural selection. Okay, and here is a pedigree. And you have an autosomal recessive. So this pedigree is telling you what the pedigree is actually showing. And so you're trying to find which of them are heterozygous. Okay, so you know the people that are affected are going to be recessive. So little a, for all the people colored in. And you're trying, again, to find heterozygous. So for these individuals here to be little a, that means each parent had to donate a little a. So each of their parents has to be a heterozygote. They're not affected because they have the big A, but they have the ability to each pass on this little a. So you can go in and look to make sure your generation 1, individuals 1 and 2 are going to be affected. This one doesn't include individual 1, neither does this one, so you can automatically eliminate those. And you don't know what this person's genotype is. You don't know what this person's genotype is. He's not affected, which means he has to have a big A. And so let's see, got the big A from here. So let's see, individual 3, or generation 3 here has to also be a heterozygote. So you can get rid of this one. And let's see, you don't know what this one is. Could be big A, or big A, little a. And you know here, this one has to be a heterozygote as well. Did I do this right? Yes. So yes, the answer should be C here. Is that clear to everyone? This is kind of an involved process. Yeah? Yeah, go ahead. I can't hear you. Generation 2, which individual? 4, right, because it's not affected, so it has to have the big A, but it did get this little a from father. So it has to have a big A because it's not affected, but did get this little a from here. Generation 4, number 4? I don't know what. Right, we don't know what this person is. Yes. Yeah, does that make sense? Yeah. Let's see. I guess you could either look at it because this is the answer that was, the correct answer in the exam. So it could be either this one or this one. Let's see. Okay. If it was this one, oh, okay. I think I understand. Let's see. It was big A, big A. I guess you just have to go under the assumption here. Because if you look back, I don't think you have any answer choices that involve 1 and 4. So if that was the case, then you'd have to go with your next best guess. So it's from elimination here what your answer choices are. I don't think she gave you the option of 1, 4 being correct. Oh yeah, she does right here. But it doesn't include the other people that are necessary. So you need to have like the best option here because pedigrees do involve a lot of assumptions and guessing. Yeah. Right, yeah. Yeah, you can't say 100%, yeah, exactly. So a lot of assuming, guessing, best answer choice, maybe not always the 100% correct answer choice, but the closest to it, yeah. That kind of has to do with the question. We got lots of questions about the one that said most likely mode of inheritance, that kind of stuff. It can be kind of ambiguous. Okay, so this is number 10 for exam 2. Only random mating in a population, no other selection pressures. The expected outcome is that, and this has to do with Hardy-Weinberg, what is the whole point of Hardy-Weinberg, is that the frequencies will stay the same. So both genotypic and allelic frequencies. That's what Hardy-Weinberg is measuring when he has all of those, or they have all of their assumptions that all of these things are staying the same. Okay, and here we have trying to figure out what the pedigree type is. So males and females equally affected, you should automatically think autosomal. So nothing with the sex chromosomes. And when there are unaffected parents, a quarter of the time you would have an affected child. So when you have unaffected parents but an affected child, that can't be dominant because the parents would have to give their dominant trait to the child, so it has to be recessive. You can also think that because if you have an unaffected parental generation, skipping generations is a trait of autosomal recessive. Okay, and here number 14, I got it started so I could explain it. There are 3 alleles in a given population. You have P, Q, and R. So you have 36% are homozygous for P, so that would be the genotypic, not the allelic frequency. 4% homozygous for Q, so again, genotypic, not allelic. And you're looking for the frequency of just R, not RR, not any kind of combination, just the one R. So you correspond here, you have 0.36 equals your genotypic frequency, PP. And 0.04 corresponds to your genotypic frequency of Q. And going back to remember, all of your allele frequencies have to equal 1. So P plus Q plus R has to equal 1. You know P because you can just take the square root of 0.36. You know Q because you can take the square root of 0.04. You're solving for R. You know it all has to equal 1. If you solve for it, you should get C, 0.2. Another pedigree. Okay, so you know the trait is not on the Y chromosome, so it's not Y-linked. This is the most likely mode of inheritance. I think this is the question I got the most questions about. and what you have to do is use your traits that you've memorized to figure it out. And here you just kind of go down the list. There's no way to just look at it and eliminate. So with autosomal recessive, you can assume that it usually is going to skip generations, so it's probably not going to be this one, but we'll keep it in the running. You can't immediately cross it out. In the X-linked recessive, you're never going to get a father giving it to a son because the father is only going to be giving the Y chromosome. So this absolutely cannot be the most likely mode. So father cannot give to son. And the same thing with the X-linked dominant. The father is giving this trait to his sons. That can't happen. So this one, autosomal dominant, that requires the least amount of assumptions in this case. So that would be the correct answer. Okay, so this is number 17. And talking about recombination here. And recombination is the source of genetic variation. And to get genetic variation, you want to cross things that are different. So you're not going to cross over between sister chromatids. So A is wrong, and E is wrong. So you want non-sister chromatids here and here. And let's see, what else? Okay, and so a recombination occurs through crossing over between non-sister chromatids. And I guess this question is also asking, does independent assortment also do this? And the correct answer, yes, independent assortment does occur with recombination because you get those alleles that shuffle differently. If we have, in this case, let me get, I guess I'll use this. If we have an allele that's kind of different here on this side, in this case, and then the next time it divides, we have an allele, I guess that doesn't really make sense. And this goes this way this time. And so this kind of weird allele, let me make this a little more clear. So this allele is going to the right this time. When it lines up again next time, it might be on the left. So this is going to create different types of gametes or different, mostly different types of gametes because this allele is going to go with this one this time, but it might go with this one the next time. There's no order, there's no direction in which each allele is, whether it's to the right or to the left of the cell. It's going to change each time because it's random, so that is going to give you recombination. Which one? Yeah. It would be X-linked recessive because the father would have whatever is recessive, and the mother doesn't, and the father only gives Ys to his son. This is most likely mode of inheritance, and that you're talking about a lot of assumptions being made, and that's where this problem is kind of tricky, is that you need to find the one that has the least amount of assumptions being made. So you're assuming then here that she would be a heterozygote. And I guess, yes, she could and potentially pass it on here, but autosomal dominant is the more likely. It is possible, there's no way to know for sure, but it's just more likely that it's autosomal dominant. Less assumptions have to be made, yeah. I can't hear you. I can't hear what you're saying. What? You can't really assume either way, because either way you're just making assumptions. The only thing that you can assume is that when it's talking about homozygous dominant, a lot of times that's lethal. And that is a fair assumption to make. So it was mentioned in the slides a few times that if you have something like big A, or an X chromosomes that even have big A, it can be assumed pretty fairly that this is going to be a lethal condition. That's something that's just known. It's just something that for some reason happens. So I guess that could be a reason why you could make an assumption a certain way or another way. The only exception to this is if you're talking about males. This wouldn't be a lethal condition. Usually you're talking about autosomes here, but this is an assumption that you could make that would be fair. But this is just a really tricky question that you just have to find out which one has the least amount of assumptions being made. because there are multiple things that could potentially work. So it's just which one is most likely. Okay, this is number 20 in exam 2, version A. So a 3-point test cross of 3 linked genes, A, B, and C. The most common would be homozygous dominant and homozygous recessive. Your least common are here. And from that information, what is the correct order of the genes on the chromosome? This is similar to a question we did before. Least common are always going to be your double crossovers. And in your double crossovers, you're looking for the gene that doesn't belong. So in this first offspring here, you have one big A and then alleles that are recessive. And in the other one, you have a little a when the alleles are dominant. So your gene order is going to be A in the middle, B, C on either side of it. And remember, genes can be read the same forwards and backwards because they're 3D things that move around in space. So C, BAC, or CAB would be correct. Okay, so you have a 2N with 16, and you're looking for these, or the number of linkage groups. And a rule of here is your linkage groups is always equal to your haploid number. That's the trick here to answering this question. You need to know your linkage groups are going to correspond to your haploid number. Here, your haploid number would be 8. Okay, and here's a really long question. So this is kind of like a step in a test, or in a 3-point test cross. So you have 3 genes, and you have mutations. So you have 3 mutations here. One for cut, one for black body, and the third for the eye color. And you do a test cross, which means you're doing your unknown with a homozygous recessive. And you determine that the eye color is in the middle. And so let's see, you use the heterozygote and the coupling. This is a bunch of information you don't really need. The expected phenotypes of the offspring resulting from a single crossover between cut wings and the eye color. So you did determine that the eye color, which is the V here, will be in the middle. And we'll say C, the cut wings is here, black body B is here, just for reference. And this is the chromosome it's on. And you can reduce this information to be really simple. So let's say this is our heterozygote. So again, this is our, let's see, I'm sorry, I did plus. So this would be our wild type. And then we'll do our heterozygote down here. And you're looking for a single crossover between the cut wings, so that would be CT, and the eye color. So you're doing a crossover here. So what would be the phenotypes? You have to I kind of like to think of it as like you're going down a road or down a train track. You start at one end, and you're going along until you find this crossing. You go down the crossing and continue on your way. So the first one would be the vermilion eye color, a black body. That would be your first phenotype. So this one here. Do the same thing with the other chromosome. You go down the road, up the crossing, and continue. And then that would just be cut wings. So that corresponds to answer A here. The cut wing mutation with your wild type eye color and body color. Your other one, your crossover, would be your wild type wings with the eye color mutation and the body color mutation. So all this information can be reduced to something pretty simple like this. Okay, and another really long-sounding question. You're looking for the recombination frequency, eye color, and wing shape. And let's see. You have a yellow mutation, and going through, your kind of recombination frequency. So you're doing a test cross. So remember, the rules of test cross here would be you are crossing with a homozygous recessive. So get rid of any answer choice where you're not crossing with a homozygous recessive. And this one doesn't make any sense because you're not going to find out anything crossing something with the exact same thing. So that can't be right. And we're doing a test cross, so it needs to be a homozygous recessive and, let's see, crossed with a heterozygote. So you can either go from there, or you could go back and look and see. Oh, it doesn't even tell you. All that you need to know is that you're crossing a homozygous recessive with a heterozygote. So that would be D. That's all you have to do in this kind of situation. In this case, both Ys here are wild type, and in this case, you have the all wild type. So you want the one that is different. You have the wild type with mutation, wild type mutation, crossed with the homozygous recessive. So this is just making sure you know what a test cross is, even though it sounds really complicated. Okay, and in this question, you're actually calculating the recombination frequency. And again, that formula is just the number of recombinants divided by the total offspring. And multiply that by 100. So that would be, in this case, 37 plus 41 divided by your total. So 80 plus 78 plus 37 plus 41. Multiply that by 100. And remember that your recombination frequency directly corresponds to your map unit distance. So that should be either 33% or 33 map units. They're equivalent. Okay, so you're given the recombination frequencies between 4 genes, and you're asked to find the order of those 4 genes. And even though there are 4 genes now, you just use the same rules that you have before. You find the biggest number, which corresponds to A and C, which means it has to be the farthest apart. So go back to your answers and cross out anything that doesn't have A and C being as far apart as possible. Oh, that one does. Okay, so these are the ones that you're left with. And then the next best thing to do here is to find your smallest number. So your smallest number here is C and B, which means those need to be close to each other. So if you have an answer choice that has C not right next to B, that can't be right. So this one, C is not next to B, C is not next to B, has to be A. Okay, and I don't think I did anything off of this page. So now I think we're in exam 3. And know all your experiments. I think that was, I'm not sure exactly what chapter that was in, but there was one chapter that had like 5 experiments. Know all of those. Okay, so you have the Hershey-Chase experiment. If the radioactive sulfur, so the sulfur here, had been found inside the bacteria rather than the phosphorus, what conclusion would have been made? And here it's testing to make sure you know the experiment and what they determined and how they did it. So remember, sulfur here corresponded to the bacteriophage's protein coat, when the phosphorus corresponded to the DNA within the protein. Remember, phosphorus is in DNA because of this phosphate backbone. And so if sulfur was inside, that would have meant the protein coat would have gone inside the bacteria. So they would have thought that proteins were the molecules that were encoding for heredity. Okay. And here's another long question that really only has one simple concept behind it. You're given a methylation table, and remember that methylation corresponds to decreased transcription. So the more methylation you have, the less transcription you'll have. And you're trying to find the one that has the most gene expression. So the most gene expression is going to have the least amount of methylation. And that's all you need to know to get the answer E. Okay, and here you're talking about DNA organization levels, and your primary level is going to be your linear nucleotides. Your tertiary level will be supercoiling. And you're talking here about the hydrogen bonds. Hydrogen bonds gives it the helix shape, and that is what secondary involves, your helix. So hydrogen bonds between the bases would affect your helix, your secondary structure. Okay, and here you're given another table. And you're asked to just find which of these 4 tubes contains human mRNA. So what you know about human mRNA is that it is single-stranded, and it has U's, not T's. So get rid of any tubes that have T's in it. So that would be tube 1 and tube 3. So now you have 50-50 chance. And then it's single-stranded, which means there is no bonding. There is no correlation between the number of A's and U's and the number of A's and G's. So looking between tubes 4, A is going to bond to U, C would bond to G, if it was double-stranded, but we know it's not. In this case, it does seem to be that these numbers are correlated, so they are bonding. There is double bonding, or bonding taking place, indicating a double bond, or a double-stranded molecule, compared to 2 here, which has A's and G's being about the same, and C's and U's, being about the same. Which those don't base pair, those don't bind in any way, so those numbers corresponding indicate that it's single-stranded. And you were told before that you are given human DNA, human mRNA, single-stranded DNA, and double-stranded DNA. So you could identify what each of these tubes did contain. Does that make sense? Yeah? Okay. And here you're just identifying this diagram here. And you can see here you have a 2' OH that is going to correspond to RNA. So get rid of anything that has DNA in it. And your purines are always going to have 2 rings on the bases. Pyrimidines always going to have 1 ring. So here you only have 1 ring. That is going to be a pyrimidine. Okay, so you are adding acetyltransferase to a sample of DNA, and what would you expect to see in the results? And that's really it. Okay, so remembering that acetyltransferases increase transcription. So get rid of anything that says that will be less transcription, or in this case, gene expression. So there will be more gene expression, not less. And then it is increasing transcription or increasing gene expression because it is making histones less positive. Remember, histones are what DNA wraps around, so if that interaction is weaker, that DNA is going to be wrapped around looser, so enzymes can come in, transcribe. So we want histones to become less positive, so not this one, and not this one, and they become less positive, causing the chromatin to loosen, not tighten. The answer here would be C. Okay, and now you have cells that are grown in culture in radioactivity, and so that all strands of the DNA molecule have radioactive nucleotides. Then you grow them up in medium that is not radioactive, and you're asked what percentage of DNA will contain the radioactivity after 2 rounds. So I think the easiest thing here is to draw it out. So let's say our squiggled DNA is radioactive, and now it's going to replicate and form 2 new strands. So now we're going to have still that radioactive old strand with a new strand that is not radioactive. Another one that is half radioactive, half not. So this is our first round of replication. Then we go to our second rounds of replication. We still have our old strand and a new strand, but now we have non-radioactive strands. This one also replicates. So we have radioactive and non-radioactive, and non and non. And this is our second round of replication. And percentage of molecules that have radioactivity is 2 out of 4, or 50%. Okay, another important fact to know here that we've mentioned a few times, that A and T involves 2 bonds, G and C involves 3. Important to know, those bonds are hydrogen bonds. So I think in here it talks about phosphodiester. That does not occur between bases. Phosphodiester is the backbone type of bond. You can think phospho is the backbone because phosphates make up the backbone of DNA. So this one can't be right. This one can't be right. That one. So only hydrogen bonds. A's and T's have 2, G's and C's have 3, and in that kind of train of thought, C's and G's have a stronger bond. So the more GC content you have in your DNA, the stronger it's bound. Okay, so do you guys have any questions before any like specific questions that you definitely want me to address during this review? Anything that you guys have thought of, found tricky? No? Second? No? Okay. I guess I just want to give you some information about the stuff in the beginning of my packet. Okay, so the first page here is a handout that was on my site since chapter 2 here. And I do, in each chapter, tell you what the main focus, I believe, is. And so in chapter 2 here, I believe the main focus is cellular reproduction, mitosis, meiosis. Pretty broad, but it's something, I guess. So here you're comparing and contrasting mitosis and meiosis. And each of these numbers then corresponds to the numbers below. So cellular division, here's number 1. I give you more details here below the table in number 1. So in meiosis 1, there's cell division in every single stage. Yeah, what's up? No, I was not, because that just happened. Do you have any specific questions? If you have specific questions, more than happy to answer. Oh. Okay. If you have any specific questions about exam 4 or exam 4 material, let me know, because I wasn't planning on going over that because that was like a week ago. Okay, so definitely cell division occurs in all types of cellular reproduction. That's the whole point, is cell division. And, okay, so chromosome reduction. That does occur in meiosis 1 and mitosis, or only occurs in meiosis 1, not meiosis or mitosis. And remember, I think I say it on the next page here, that meiosis 2 is basically just mitosis. You're just going from 2 cells to 4 cells. There's not a whole lot of difference in what happens, except you're working with the haploid number. And in that vein, the haploid number occurs in meiosis 1. So after meiosis 1, you're haploid. Nothing else happens in meiosis 2 except you go from 2 cells to 4 cells. Okay, and genetic variation only occurs in meiosis 1. Meiosis 2, there is no crossing over. You're just, again, making 4 cells out of 2. So again, same thing, crossing over only in meiosis 1. Homologous pairs are only present in meiosis 1. You're down to the chromosomes in meiosis 2, and you're splitting them into their chromatids. And so there are no chromatids present in meiosis 1, but there are in meiosis 2 and mitosis. So use this table to just kind of know the information about each stages of cellular reproduction. And again, like I said, each of these numbers corresponds to more information below here. So all that information. And here is kind of a diagram mapping out the different, this is meiosis, so this is meiosis 1, starting from metaphase going to 2 cells here. So you have your homologous pairs each here. And they are separating in anaphase. And now you have the haploid number. So remember, count your centromeres. You have half the amount of centromeres at the end of meiosis 1 than you did in metaphase, so you go from diploid to haploid. Compared to meiosis 2, you keep the same number. So you start with the same number of centromeres, you end up in each cell. You're just separating these chromatids. This is a good thing to use if you're having trouble counting chromosome numbers and DNA molecules. Use these tables and also try to create your own. It's the best way to learn. Okay, and a minor concept within chapter 2 was the prokaryotes and eukaryotes, differences, compare, contrast, that kind of stuff. Okay, and then chapter 3, the main focus was Mendelian genetics, so that would be your Punnett squares, your rules of probability, all of that stuff. And so some key things to remember here, that there are, each organism has 2 alleles for each character. So even if there are 5 alleles in a population, remember each organism is only going to have 2 of those. And this, again, I give you the definition of the law of segregation here, that these 2 alleles are going to separate, so each gamete will only get 1 of those 2 alleles. Compared to the rule of independent assortment, which says that when they segregate, it's completely random. So allele 1 will separate no matter what allele 2 is doing, and all that stuff. And here's the probability rules. Remember, and is multiply, or either add. Sometimes you have to kind of read into what the problem is asking. Sometimes it clearly says and, or. Okay, and here I give you an example problem using simple Punnett squares, and then the key thing you have a 50% chance of getting a son versus 50% chance of getting a daughter. So always remember, half chance of daughter. When you do your Punnett square here from the information I gave you, it is a half chance of having fabulousness. So when you multiply them, because you want fabulous and daughter, even though it is assumed here, it's a 1/4 chance total. Okay, so chapter 4, the main focus here were the sex-linked genes, and use the same rules as you did for the autosomes when using Punnets and probabilities. Just remember that the male-to-female ratios will be different, and that's the key thing to look for, especially in pedigrees, is that your ratios will not be the same for males to females. So here are some minor concepts here were the non-disjunction sex determination. And remember here, the ZW, that one was the one where females are the heterozygote, or the heterogametic, and males were the homogametic. All other ones, the males are the heterogametic. And here's your non-disjunction. And know what each of these phenotypically present as well as genotypically present. So here, your XYY, those were your super males. So they will be phenotypically male, usually more aggressive, maybe kind of a little bit bigger. Your triplo X, kind of self-explanatory, you're going to have 3 X's. Most of the time, if you have something going on with the X's, you're going to have an infertile female. Turner syndrome, all of these, I believe, are still going to be female. And so just make sure you know all of the different characteristics of these, that stuff. Okay, and here's chapter 5 material. The main focus here will be all of those exceptions to the observations. Chapter 5, pretty much just vocab. And so here's kind of an example type of problem to help you maybe remember, because it can get confusing. You have incomplete and codominance. Incomplete dominance, let's see. Actually, let me not do that there. Let's see. Codominance is, you can think you're like co-manager, co-something. You're both equal. You're both going to be equally represented. You're both equals in the situation. And that's how you can remember codominance. You can also remember it in this picture represents codominance because this is a platypus, in case you can't tell. I don't know if you can. But this is a duck at the top and a beaver here. So if a beaver and a duck have a baby, it's like a platypus because it has like the tail and the beak. That's just, yeah, that's a way to remember codominance. When incomplete dominance is where neither one is represented fairly, I guess you could say. It's a complete blending. So if you blend and a fork and a spoon have a baby, you're going to get a spork because you blend. You're neither a spoon, you're neither a fork, you're blended into a spork. So this would be incomplete dominance. I hope that there's a question on this and you think of sporks during the final. Okay, and here is a formula to remember. You probably can figure it out multiple ways. This is just the formal way of remembering how to find out the number of genotypes that are possible when you're given the number of different alleles. So if you know there are 5 alleles in a population, you use this formula here to determine how many genotypes are present. That is kind of a detail that I'm not sure if she would put on the final, but she did say broad topics, but don't gloss over small things. If you're looking for more detailed information, definitely reference my other exam reviews. And here I do give you some of the ratios. So your 9:3:4 ratio was your recessive epistatic ratio. Your 12:3:1 ratio was your dominant epistatic ratio. So be able to recognize those because it'll make the exam go a lot quicker. Okay, and the difference here, another key thing, is your difference between sex-influenced and sex-limited. Your sex-influenced will be that the trait is dominant in one, recessive in the other, when sex-limited will be 0 penetrance in one, but not the other. So I think the example in the sex-limited was the chickens and the roosters. So the chickens, the female version, is never going to have, I think, those crazy featherings that the males could have. So never going to happen, sex-limited to only involve males. Sex-influenced, I believe, talked about baldness, in that it's a dominant trait in males, recessive in females. So even though it is one trait, it has different effects. Okay, where am I? Okay, chapter 6, that was all pedigrees, only pedigrees. If you know what each pedigree traits are, how to figure it out, how to find it, you're fine with chapter 6. So I give you what each of these pedigrees are usually involved or are involved in. Remember your X-linked and your Y-linked, those sex chromosomes, you're going to have different male to female ratios. If your males equal your number of females, it's going to be autosomal. But don't always rely on that. If you're taking the final and you count the number of females and count the number of males, don't just base it on that. Do a little bit more information, or find out a little more information. Okay, remember consanguinity, that is involved in incest. So if you see those double lines between people, that I would safely assume that it would be autosomal recessive. Let's see. Yeah, okay. And it was mentioned in the slides that you can assume that homozygous dominance is usually lethal. But again, don't only base your answer on something like this. And the best thing to do, the best way to get the right answer in pedigrees is to rule out other things before you choose one thing. So just because you did it and it makes sense and you think it works, check the other ones just to be sure. Okay, in chapter 7, linkage, the favorite chapter I know. The key thing here is that crossing over is an event and recombination is a process. So they are two separate things. Crossing over is something that actively happens when recombination is just the result of what crossing over did. So they are related, but they are separate things. Let's see, so your typical 9:3 to 3:1 ratio here can be altered to be a 1:1 to 1:1 if there's independent assortment, or a 1:1 if there is complete linkage. So again, just ratios to know here. And again, remember your percent recombination equals the number of map units that are present. And so an example of that, 12% recombination means those genes are 12 map units apart. And here's your formula for determining percent recombination. Okay, and I gave you the basic steps of determining map distance between genes. So you find the total number, oh, sorry, you can't see that. You find the total number of progeny, you find the total number of parental recombinant, and you calculate the recombination frequency. Let's see, and that would be your map distance, because remember, percentage recombination equals your map units. And then the basic broad overview of how to do a 3-point test cross is you want to ID all of your offspring. So your non-recombinant will be your parentals, that will be your biggest number. Your double crossover progeny will be your smallest number. And then you compare these two guys together and determine the one that's different or doesn't belong. So if you're comparing these to be your parental, your P generation, and these are your double crossover, your smallest number, determine here that C is the one that's different. That means that one's in the middle. And I would recommend rewriting, if you're given a table, all of those in the correct order, because I've done it multiple times where I don't, because I don't think I have to, and then I mess up. And the last thing you need to do is, with the correct gene order, identify those single crossover offsprings, and then calculate the recombination frequency. You'll know when the single crossover offsprings correspond because they'll be similar numbers. They won't be more than 2 or 3 numbers apart, so like 31, 27, 17, 15, something like that. And remember, when you're determining the recombination frequency, to add your single crossovers with double crossovers. So in a case where you just have, I don't know, just making it up, and you have double crossovers, those would be your double crossover, but then, let me add. And then you have your single crossovers. Just one here and then another one on the other side. You find each of these, and you're going to calculate the recombination frequency. So let's say you're doing this one first. You find the recombination frequency here, and when you're adding the numbers that correspond to this type of crossover, so let's say it's something like 13 and 10 or something, that was the number of offspring that correspond to this single crossover. When you're adding these numbers, always make sure to add your double crossover progeny as well, because this crossover did take place in the double crossover progeny. I think that's the biggest mistake that people make, is that they don't remember to add the double crossover numbers with your single crossover numbers. So make sure you add, if it's something like 2 and 5 or something, to add that here. Because this did take place, even though you're only looking at this one, it did take place in the double crossover. Same thing here, always add your double crossovers to your recombination frequencies. And then chapter 8 here, this was populations, especially Hardy-Weinberg, if not exclusively Hardy-Weinberg. Okay, and so I give you all of the formulas that you need. So if you're finding the frequency of homozygous dominant, remember that would be your genotypic frequency, not your allelic frequency. You just take the number of total individuals that have that genotype divided by n, which is every single individual. Same thing if you're finding the homozygous recessive or the heterozygote. And now if you're trying to find the allelic frequency, which would just be 1 A, 1 big A, 1 little a, whatever you're trying to find, you take the frequency of your homozygote for whatever you're looking for, and you add that to half of your heterozygote, because you're only wanting that 1/2 of the heterozygote genotype. Same thing for Q. So this, remember, is your genotypic frequency, and this is your allelic frequency. Genotype, you have 2. Allelic, you have 1. In here, I give you your Hardy-Weinberg assumptions. Kind of, maybe. Yeah, here are all your assumptions. Populations are large, random mating, no mutations, no natural selection, no gene flow. Gene flow, remember, is migration. And here is your Hardy-Weinberg formula. So your frequency of allele A is P, or just the dominant is P, recessive is Q. And here is your genotypic frequencies. And remember, all of this has to equal 1. So this is the formula definitely to remember. If you only remember one formula, make it that one. Your P squared plus 2PQ plus Q squared equals 1. And this is the chart, again, I would commit to memory. What happens when you increase variation, or what you have to do to increase variation within populations, between populations, all of that stuff. And then remember, genetic drift is different from gene flow. Genetic drift decreases because it has small populations, which are examples are the founder effect and genetic bottlenecks. That was talking about the panthers and the ladybugs on the island, I think, in the PowerPoints. But definitely this chart. This is probably this and something where you have to use this equation, or what you'll be asked from chapter 8, I would assume. Okay, and then definitely changing focus. Chapter 10, you're just kind of getting the basic introduction into DNA, and know the difference between a nucleotide and a nucleoside. And I remember I said this was wrong, flip these two things, cut pi, remember that, don't remember what I wrote. And you're talking about hydrogen bonds here. And here's your levels of DNA structure. Know the difference between ribose, deoxyribose, because that's the difference between DNA and RNA. Remember, it's your 2' OH here, your lack of 1 here. Remember, deoxy means without oxygen, so you don't have an oxygen at that 2' carbon. That's the broad aspects of chapter 10. Then chapter 11, that would be structure, especially talking about those histones, which we did a question on. These are your 5 types of histones. Remember that you have 5. And in your nucleosome core, you have 8, so everything except H1, and you have 2 of each. Your H1 forms your histone that links the DNA to your nucleosome. And remember, histones have an overall positive charge because DNA has a negative charge due to the phosphate group. And then chapter 12, replication. If you memorize and understand what you can't see right now, this structure, this diagram, then you should be good to go for DNA replication. Know your leading and lagging strands. I list all of the enzymes that you should know. Know what happens when they are working. Know what happens when they're not working. Directionality of things. So it gives us the template strand here. So we have a 5' template strand here, 3' here, which means that the replicated strand has to be moving in an anti-parallel fashion. So if this is the 5' end of the template strand, then this has to be the 3' end, if you can see that. The 3' end has to be here, on this end of the strand, to make it anti-parallel, when over in this end would be the 5' end, back over there. Same thing with the bottom strand here. If this is the 3' end of the template strand, then that means this has to be the 5' end of the new strand. And back over here will be the 3' end. And know the leading strand goes towards the replication fork, the lagging strand moves away from the replication fork. Ligase is never going to do anything to the leading strand. That's only something that works from the lagging strand. The difference between DNA polymerase 1 and 3, although they both have error-proof capabilities, or proofreading. Anything else? I think that's it. Okay, and then chapter 13 was talking about transcription. And you have your 3 stages: initiation, elongation, termination, your central dogma of biology. And your minor concept here would be the difference between prokaryote and eukaryote transcription. Also make sure you know the termination mechanism, especially in prokaryotes like the row, dependent, independent. And then here is what you had your last exam on. So chapter 14 would be modifications to mRNA, so going from pre-mRNA to mRNA, especially the difference between prokaryotes and eukaryotes. Eukaryotes need all these modifications like the poly A tail, the 5' cap. Prokaryotes don't. We also have all these introns we have to get rid of. Prokaryotes don't. And this is also why prokaryotes can use operons, because if they have one operon here, they can just code for all of these different proteins. But we have so much modification needed to take out introns, add things, all that stuff. We can't really use operons. There's too much junk in the way. And then chapter 15, this was translation. And here, again, if you kind of understand the process of this diagram, you should be good to go. Here's your 4 steps of translation: charging, initiation, elongation, termination. Charging involves your tRNAs. Initiation is just the units all combining, so the large and small subunit of the ribosome attaching to the mRNA. And then elongation is actually when you start to grow your amino acid chain. So remember, the first amino acid is going to bind to this P site, which will be in the middle, move to the E site, and then exit, recharge again, and eventually get recycled through over and over again. Remember that your tRNAs are binding to mRNA, not the ribosomes necessarily. Their anticodon region is binding to the mRNA. They are using the sites in the ribosome, but they are binding to mRNA. And remember, they're using their anticodon region to bind. They're using their 3' ends to attach the amino acid. And their unique modified nucleotides are what code for which amino acid binds to each tRNA. So each tRNA only binds to one specific amino acid. It's not like every tRNA can bind to every single amino acid. And it determines that based on those weird nucleotides like modifications on G, that kind of stuff. So they are specific. And here is a table to try to help you with the operons, because I know that can be difficult. So each column is a different operon. You have your negative inducible, which is, remember, is your lac operon. The regulatory gene will be a repressor. And for transcription to occur, you have to prevent this repressor from binding to the operon. And that is done by allolactose binding here. So this is often involved in catabolic pathways, so when you're breaking something down. And this repressor is in an active form because it's repressing transcription, which is what's usually happening in an inducible operon anyways. When a negative repressible operon, you have this negative again, so you're coding for a repressor. Repressible operons are usually on, so this repressor must usually be in an inactive form, or else transcription would usually be off. And it's usually involved in anabolic pathways. So definitely know which ones are involved, catabolic, anabolic. When you see positive, you can think activators. And inducible, remember, is usually off. So if the regulator protein is an activator, it has to be in an inactive form unless something happens. And the opposite is true for a positive repressible. You're coding for an activator. The operon itself is usually on, so that means that activator must usually be active and working. Know the difference between inducer, co-repressor. So an inducer is going to activate the activator. A co-repressor is going to help the co-repressor, or the co-repressor is going to help the repressor work. Okay, and here is a diagram of the lac operon. And here's your lac Z, Y, and A genes. Know which each of those correspond to. So beta-galactosidase, permease, transacetylase, but don't worry too much about the last one because we don't even know what it does. Know that there's other ones too, like lac I, lac O, and there's the allolactose, which is the inducer, all of those different factors in there. And the last chapter that I talked about here, actually I have more pages. Here is chapter 17, which is eukaryotic transcription, and this was kind of a limited chapter in information. There's only a few things to know, and it's that has to do with enhancers and silencers. Kind of self-explanatory. Enhancers are going to enhance transcription. Silencers will silence transcription. And let me see. And then I have it up here. And this scary list is all of the mutations that you need to know what they do in chapter 18. So all of these are mutations here in this column, and then all of these are the repair mechanisms in the other column. So know what each of them do, what happens when each of them occur, and then how each of these will try to fix each of them. And know for translesion repair that this is a last resort mechanism, because translesion repair is only used to just bypass errors. It's not going to fix anything. It's not going to replace anything. It involves a polymerase that comes in and takes the place of regular DNA polymerase, because regular DNA polymerase you can think of as a perfectionist, and once it sees an error, it can't move past. It's not going to keep going until that error is fixed. So translesion polymerase comes in and says, no, that's no big deal. We'll keep going and just ignore that error. So this error still occurs, which means mutation is going to happen. So this is an absolute last resort mechanism. In the last chapter, chapter 19, were all of the techniques. And I list all of these techniques that she mentioned, which was electrophoresis, the blotting, cloning, PCR libraries. Know when you would use which libraries, which of these things involve mRNA or DNA or protein. And let's see. Okay, so and here I say just expect that in each exam she didn't cover every single topic, every single concept. So if you know a concept was pretty big and you didn't see it in an exam, it's probably going to be on the final. Okay, and I think we have 15 minutes left, so I'll leave that open for questions, or if you want to see something again or have a concern of any type. Yeah. Okay, yeah. Do you know what chapter that was in so I can flip to it? That was involved in the sex determination, so kind of earlier. Okay, so non-disjunction, you can think the chromosomes are not going to be disjoining. I guess you can think of it that way. So some gametes, some cells are going to have too many chromosomes or too few. When it comes to sex determination, you were given 4 different types of non-disjunction. Turner syndrome is phenotypically female, and I think they only have one X. So they would be XO, X nothing, basically. So they're usually sterile, usually shorter than average, and definitely know this genotype of them. Triplo X, self-explanatory, 3 X's. They're usually sterile, I believe, and they're usually, I think, larger than average women. XYY, that's going to be your super males. So those are the ones that are more aggressive, larger than average males. And your Klinefelter syndrome, I believe that is phenotypically male. Can anyone verify that? Yes, I'm nodding. Yeah? I think it's phenotypically male. Do you remember what the genotype is? Thank you. I wanted to make sure. Okay, so usually you're going to be XXY in Klinefelter syndrome. So this means you have an extra X than you need. These males probably would be, they probably wouldn't be sterile, but I think they have secondary female sex characteristics. They might have reduced male sex characteristics. But are you talking more about like the nullosomes, monosomes, that stuff? Okay. Number 27. Okay. So we have a 2n number of 24. Non-disjunction event occurs during meiosis 2. Okay? And we have 1 egg from the round of abnormal meiosis is fertilized. What would you expect to see in the offspring? Okay, so there is a slide in the lectures that actually goes through and gives you what happens in each of the non-disjunction events during mitosis, meiosis 1 and 2. So let's see. So you have your 2n number, which is usually 24. And in meiosis 2, that is one that we talked about in class that said that's probably going to be not as big of a deal as the other ones. If you're going to pick a non-disjunction event to occur somewhere, you want it to occur in meiosis 2, because you're always going to have half the number of normal gametes. So here, if you can remember this, or you can go through the entire process, but you can remember that in meiosis 2, you're going to have half the number of normal gametes. So from this, you can go in and get rid of any of the ones that say they would not be normal. So you're going to have half of them be 24. And then the non-disjunction event that occurred before, you're going to have one that is -1 and one that is +1, because something happened where it got pulled to one side got extra, one side got less. So a quarter of them are going to have one less, a quarter of them are going to have one more in this case. And that's see. I think that if you go to the slide in the lecture that explains that, it would be super helpful. Yeah, because I think it's easiest, honestly, to just memorize these types of things as opposed to having to draw out what happens in each stage and figure it out. So if you memorize what happens in meiosis 2, 1, mitosis, and all that, it'll be a lot faster. Yeah, any other questions? Yeah, go ahead. Everything always goes 5 to 3. There's no change in anything. Yeah, it's always going to grow 5 to 3, but the way you read it might be 3 to 5, 5 to 3. Right, because of the 3' OH. So if there is no 3' OH, no addition can happen anyways. So always growing 3 to 5, but you can read it either way. Mm-hmm. Yep. Anything else? Basic question. Yeah, the 3' OH on the pentose, that's what actually allows the base to come in and grow. Yep. Yeah, no problem. Anything else that you guys have questions on, concerns on? Let me go back through anything. Any like specific? I know some of the problem-based questions can be difficult. Any of the math, anything like that? No. Okay. Let me see. Okay, so to clarify, then what we just talked about, because I know this picture is kind of hard to see, but you have a 5 to 3' end and a 3 to 5' end because they're anti-parallel. You always need DNA to have that. It can ever be, if you have 2 strands here, it can never be 5 to 3 and 5 to 3. That can never be what's happening. It always has to be 2 strands, one 5 to 3, the other 3 to 5 to bond. But when each of these separate and grow, they're each going to grow in a 5 to 3' way. So it can either be growing 5 to 3 to the right, 5 to 3 to the left. That doesn't matter as long as you're just growing towards the 3' end, because back up here is the 3' end, has that OH in both RNA and DNA, which is, probably the most important part of the nucleotide is this 3' OH. Because if you don't have this 3' OH, you can't add any bases. And this is the technique that's used in the Sanger method, which is in the last chapter of the techniques, where if you use nucleotides that don't have this 3' OH, you're not going to get any more replication because it's just going to stop. You need this 3' OH to bond with the phosphate that's coming in to just synthesize and create that phosphodiester bond. If this is just an H, it's not going to happen. So this is why it's 5 to 3. Is everyone comfortable with what each of the enzymes do in replication? Everyone feel good about that? Okay. Any detail questions? Anything? I'm surprised that we have any questions. Yeah. 100 multiple choice questions. Yeah. She didn't say whether to focus more on one chapter or the other, so I would assume a fair distribution. I'm not sure. I say at the beginning of the review how many chapters there are, or I guess so you have chapters 2 through 8, 10 through 19, and chapters 25, so I would assume a fair distribution between however many chapters that is. So you can figure that out, how many questions per chapter, probably. Yeah. Make sure you bring a calculator. Don't not bring a calculator. And your ID, too. Anything else? Any last questions? No? Yeah. Oh, 80. Yeah, and before you leave, either leave it where you are or put it to the end of the table. Make sure I'll collect them, definitely. Yeah, make sure you bubble in, write 80 at the top there. If you have any questions, I'll be here till 4:30, so just let me know.